Hi, today we are going to talk about mitosis. Mitosis is one form of cell division. It is also one of the most fundamental processes in biology, and it is probably happening inside your body right now.
It is crucial for growth and development of our body, from a single cell to a small baby and finally becoming an adult. Mitosis is always occurring to repair and replace worn-out tissue so that our body heals after an injury. The regulation of mitosis or cell division is extremely intricate because if not, tumor will develop and many other processes in your body will go haywire. Thus, a deeper understanding of mitosis will allow scientists to discover more about our body growth, cell repair, as well as cancer. Mitosis is so extensively studied. Now, let us take a glimpse at the overall process of mitosis. The ultimate goal of mitosis is to create daughter cells with the same amount of chromosomes, which contain our DNA. Therefore, the cell must duplicate the chromosome first. Mitosis can then distribute the chromosomes equally into the two new daughter cells. As we can see from this diagram, mitosis starts once chromosomes are duplicated. There are a total of 4 stages in mitosis: condensation, prophase; alignment, metaphase; separation, anaphase; and cell splitting, telophase and cytokinesis. One round of mitosis will give us 2 identical daughter cells that have the same number of chromosomes as the original cell. In order to make sure that the chromosomes are properly distributed, it is critical that the third step is carried out correctly. And thus, the focus of our presentation today will be the third step, anaphase. During anaphase, sister chromatids are separated and equally distributed into the new daughter cells. It involves the work of many thread-like structures and motor proteins. Let us go through some of the important terminologies. Do not worry, there are only 2 important ones. Firstly, we have the mitotic chromosome. This structure is nothing but DNA in their highly coiled and condensed form. Mitotic chromosomes have 2 sister chromatids because of the duplication event earlier. The 2 sister chromatids are attached together by a protein called kinetochore. Secondly, we have the microtubule. The microtubule is a thread-like structure that will attach to the kinetochore of our chromosomes. The job of the microtubule is to shorten and pull the sister chromatids away from each other and towards opposite poles. However, when we zoom in further, the microtubule actually comprises of many small dynamic subunits capable of assembly and disassembly. This is what happens when the subunits are assembled. As you can see, the microtubule is actually being formed and elongated. So, when the microtubule shortens to pull the sister chromatids away, its subunits are actually being disassembled. There are a total of 3 types of microtubules involved in anaphase: astromicrotubule in blue, kinetochore microtubule in purple, and overlapping microtubule in pink. Now that we know what is a chromosome and what is a microtubule, we are fully equipped. Let's look at the actual anaphase. There are 2 steps in anaphase: anaphase A and anaphase B. In order to understand this dynamic process, let's take a look at this physical model of a cell undergoing anaphase.
Let me orientate you around the model. Again, we have 3 types of microtubule: blue astromicrotubules, purple kinetochore microtubules, and pink overlapping microtubules. Blue astromicrotubules are attached to the cell cortex that is not shown. Purple kinetochore microtubules are attached to the chromosomes at the kinetochore. Pink overlapping microtubules have an overlapping region here. All these microtubules originate from this microtubule organizing center, each containing a pair of centrioles perpendicular to each other. You do not have to worry about these terms. Are you ready? Let's see how the two-step anaphase is carried out. Anaphase A is the separation of the sister chromatids. The purple kinetochore microtubules will shorten as the subunits disassemble, thus pulling the sister chromatids away from each other and towards the opposite poles of the cell. Next, we have anaphase B. The pink overlapping microtubules will slide over each other, pushing the two poles of the cell away from each other. This helps to elongate the cell for easier splitting of the two daughter cells during the last step of mitosis. Can you see that the sister chromatids of the chromosomes are now equally distributed to the new daughter cells? Anaphase is now complete.
In summary, these are the important points about anaphase that we have learned today. All these dynamic processes are not possible without the microtubules. Anaphase occurs in two steps: anaphase A and anaphase B. Anaphase A involves the separation of sister chromatids by the kinetochore microtubules, while anaphase B involves the sliding of overlapping microtubules to elongate the cells.
This is the end of the presentation. I hope that you have learned something new about anaphase and mitosis today. Thank you for your attention.